afternoon, everyone, and welcome back to the Genomics England Research Seminar Series. And what has proven to be quite a wet start to the week, but I think it's brightening up a bit now. But luckily, we've got a really exciting set of talks to kind of brighten up everybody's day. And these are in recognition of the fact that it's Gynecological Cancer Awareness Month. But before we get to the talks, I'll just quickly introduce myself because I'm a new face. I'm Keris. I started at GEL back in July, so I've been here for 2 months now. And I'm a scientific manager in the research management team. My role is to work closely with the research network and promote data use of the data in the NGRL. But before we just jump into the science, a couple of housekeeping bits. So the seminar is being recorded and you will be able to catch up on a later date on our YouTube channel. The seminar will last around about an hour and consist of 2 20-minute talks. After each talk, there'll be a Q&A session. So please use the Q&A function at the bottom of your screen on Zoom to put your questions in rather than using the general chat. Just means we can keep track of them a bit better. So if we go to the next slide, we'll go through what's coming up today. We have 2 brilliant talks coming away from Aliah Hawari and Kramer, which are both linked to gynecological cancer as per the fact that it's the awareness month. But just before we get into those talks, I'm introducing the speakers to you. I'll just quickly hand over to Emily Perry, who's just going to take you through some exciting training that we've got coming up.
Yeah, so if anybody is a registered user of our research environment and you want to kind of get a bit more practice, get a bit more skills, get better at using the research environment so that you can get more out of your research, please register and come along to our in-person training day, which is being held at our offices in Canary Wharf. You can attend in person. We think it's best if you attend in person, but you can also attend virtually if you're not able to travel. It's going to be full on, hands on. There's going to be practicals, demonstrations, exercises. Just to point out, it is only open to people who are registered research environment researchers. So if you're not one of our researchers, then you won't be able to attend. But if you scan the QR code that's on your screen right now, or if you just search on the Genomics England website under events, you should be able to find it and register there.
Brilliant. Thank you so much, Emily. And just finally, before I introduce the last speaker, just to let you know that our next research seminar will be on the 29th of October at 2 p.m. As normal, and speakers are currently to be decided. So without any further ado, I'll introduce our first speaker, Aliah Hawari, who is a computational biologist at Manchester Cancer Research Centre. She's working in the field of cancer genomics and data science. And Aliah will be talking to us about mutational signatures and evolution trajectories in ovarian cancer. So take it away, Aliah, if you want to share your slides.
Yeah. All right. Thank you, Keris, for the introduction. Thank you, GEL, for the invitation. And thank you for everyone for joining. So as advertised, I'm Aliah. I'm a computational biologist at the Manchester Cancer Research Centre. My work primarily focuses on the analysis of whole genome sequences. And in my research, I examine the landscape of mutations, identify mutational signatures, and investigate the chronological order of events in tumor development. So recently, I've applied these methods to ovarian cancer, which will be the focus of today's presentation. So I'd like to begin by providing some context on the data set used for this analysis. So the data comes from Genomics England and encompasses 383 epithelial ovarian cancers. This data set includes 6 histological subtypes. High-grade serous cancer is the predominant subtype, but we also have endometrioid adenocarcinomas, clear cell carcinomas, mucinous carcinomas, and low-grade serous cancers represented. Looking more closely at the serous cancers, we find that they are primarily high-grade, and among the non-serous types, there's a higher distribution of grade 3 cancers. Regarding staging, we see regarding stage 3 is the most prevalent across all types. So you'll see that I was talking about genomic orders or the orders or events of the genomic aberrations in the data set. So to determine the order of genomic events, I adapted a placket-loose mixture model, a method developed within our lab. This model helps us to estimate the likely sequence of copy number changes and mutations in tumor development. So it is a combined approach model with packet-loose ranking method and also a mixture modeling. So the key advantage of using a mixture model is its ability to extract distinct ordering profiles within the population or cohort. This enables us to identify subgroups with potentially different evolutionary trajectories. And the packet-loose model was used to order the events by sampling from all possible tumor phylogenies across the entire dataset or cohort, about 1,000 iterations. So this combined approach allows us to identify distinct patterns of event ordering and then predict the most likely order of events within each subgroup. So each subgroup of samples would have similar patterns of genomic event ordering. So using this placket-loose mixture model, I identified 4 distinct evolutionary profiles within the ovarian cancer dataset. There's colorful bands here that you see here depict the relative timing of genomic events for the ovarian dataset. The genomic aberration events are ordered early to late by the median value of the relative timing estimate. And I've named it DN1, DN2, DN3, DN4, DN4 de novo. So genomic aberrations included in the analysis were enriched copy number aberrations, such as gains, losses, homozygous deletions, and mutations, which is colored in red, blue, purple, and green. And each of these unique trajectory or each of these unique profiles is characterized by a specific chronological ordering of these genomic aberrations. So the first Evo type or profile, which I've designated as DN1 here, is loss-driven. 42% of the genomic alterations in DN1 are loss of heterozygosity events. Moving on to Evo types DN2 and DN4, we observe some interesting similarities. So at first glance, these 2 evolutionary types appear quite alike. So both DN2 and DN4 are characterized by a more balanced distribution of genomic events. But specifically within each of these Evo types, we see roughly equal of the blues and the reds, so roughly equal copy number gains and losses. So these balanced profiles contrast with the loss-driven pattern that we observed in DN1. Now, DN3 stands out with a markedly different profile compared to the other Evo types. So DN3 is mutation-driven, with remarkable 55% of its events being mutations. What's particularly noteworthy is that these mutations tend to occur early in the evolutionary trajectory. And in addition to this, DN3 is characterized by having the lowest number of copy number losses compared to the other Evo types. Also, DN3 completely lacks homozygous deletions, whereas the other Evo type shows some presence of these events, albeit at very low levels, so it's less than 5%. And so earlier I've shown that our cohort is predominantly composed of high-grade serous ovarian cancers. So interestingly, high-grade serous ovarian cancers are primarily distributed into the 3 out of the 4 identified Evo types, so DN1, DN2, and DN4. And in contrast, when we examine Evo type 3 or DN3, we find that this subgroup is mainly composed of endometrioid, clear cell, low-grade serous, and mucinous cancers. Same pattern or same pattern images for grade and stage. Now let's look at some of the genomic events in these 4 profiles. I would highlight the type of genomic events as such, like this is in green in mutation for TP53. And in the square brackets would be the prevalence of that event in that specific Evo type. So it would be the percentage of samples with that event in this subgroup here. So let's start with TP53 mutations, so which are known to be prevalent and typically occur early in ovarian cancer tumorigenesis. So our analysis confirms this pattern across all 4 Evo types that all occur quite early. In DN1, DN2, and DN4, we observe that TP53 mutations occurring early in the evolutionary trajectory, consistent with previous findings in ovarian cancer. However, if you see here, DN3 presents a distinctive profile, while TP53 mutations still occur early when it's present, their prevalence is markedly lower in this group. So interestingly, DN3 is primarily composed of endometrioid ovarian cancers, and this low prevalence of TP53 mutations in DN3 aligns with previous studies suggesting that endometrioid ovarian cancers may follow a different pathogenic pathway compared to the other ovarian cancer subtypes. Now, we observe a similar trend for the loss of a key region on chromosome 17, which harbors important genes such as BRCA1, CDK12, ERB2, NF1, and TP53. So in vivo types DN1, DN2, and DN4, this loss occurs early in the evolutionary trajectory. However, again, DN3, we again see a divergent pattern. Not only does this loss occur much later in DN3, but it also shows a much lower prevalence. Then next, we examine the occurrence of whole genome duplication events across our evo types. So a consistent pattern emerged. So in evo types DN1, DN2, and DN4, we observe that the whole genome duplication tends to occur relatively early in the evolution trajectories. DN4 with the highest prevalence. However, in DN3, once again, distinguishes itself from the other evo types. In DN3, not only does WGD occur later in the trajectory, but it also shows a notably low prevalence. ARID1A mutations. So ARID1A mutations are common in clear cell ovarian cancers and is associated with shorter progression-free survival. And in our analysis, ARID1A mutations are prevalent in DN3. That includes the majority of the clear cell cases. And literature suggests that PIK3CA mutations are rare in ovarian cancers, except in endometrioid and clear cell types. PIK3CA amplification, on the other hand, is common and associated with resistance to taxane and platinum-based chemotherapy. Our findings align with this. PIK3CA mutations are substantial, to the 3%, but only in evo type 3, consistent with its endometrioid composition. PIK3CA amplification is highly prevalent in evo types 1, 2, and 3, but not in evo type 4. About evo type 4, a striking feature of DN4. So all DN4 samples show amplifications of a chromosome region 19, from 19q11 to 19q13. And this region containing CCNE1 and AKT2. So this amplification is absent in all other evo types. CCNE1 and AKT2 are associated with therapy resistance. So it's typically seen in 15 to 20% of high-grade serous ovarian cancers. And DN4 here represents 21% of our cases, mostly high-grade serous. Now, DN2 here distinguishes itself from the other evo types with a complex BRCA alteration pattern. DN2 is unique among evo types in showing both gains and losses of BRCA1. DN2 is unique among evo types in showing both gains and losses of BRCA1, but it's also the only evo type with BRCA2 mutations. Now, to provide further molecular context to the evo types that extracted mutational signatures. Now, these signatures were extracted based on the COSMIC catalog from the Sanger Institute and using SIG Profiler tool. We analyzed 5 out of the 6 available variant classes, so copy numbers, single base substitutions, insertion deletions, structural variants, and also double base substitutions. And then clustering samples by evo types reveal interesting patterns in mutational signature activities. DN2, DN1, and DN4 here clustered together, while DN3 remain distinct. So note that there are more signatures extracted, but for this talk, I'm showing this once for clarity. So what you see here, each of the vertical bands here represents samples in the subgroup, and the subgroups are now clustered within DN2, DN1, and DN4, and DN3 respectively. And 7 HRD associated signatures across 5 variant classes were examined. And here HRD signatures, ABS13, ID6, CN17, SB3, and SB5. And some key observations. So HRD signatures are elevated in the N2, the N1, and the N4 cluster, but not in the N3. Now, the N2 here shows the highest enrichment of HRD signatures, if you see here, and it's particularly for ID6, which is here. So the N2's unique BRCA1 alteration pattern and BRCA2 mutations correlate with this enrichment, the pattern that we see earlier when I was talking about all the different profiles. So now these findings suggest a spectrum of HR impairment within the N2, the N1, and the N4 cluster. The N2 potentially represents a BRCA-related HRD trajectory, and the N1 and the N4 showing varying degrees of HRD signatures activity. So this spectrum of HRD characteristics across evotypes may have an implication for understanding tumor evolution and treatment responses. I've also done a Kaplan-Meier curve that shows survival between the N3 and all other evotypes. So we can see by eye, there are some differences in the curve, albeit not significant. The N3 a bit better than the other all evotypes. So to conclude, our SBS, our analysis of the 383 ovarian cancers reveals two main groups. So the homologous recombination deficient and homologous recombination proficient. And some key finding is that the enhanced HRD signature activities is in the HRD group as compared to the HRT group. And within the HRD group, we identify a cohort with enriched HRD signatures and distinct genomic ordering patterns, which is the N2. And we see some elevated in HRD signatures, which is the N4 and the N1. And we see that the N2 and the N4 has a pattern of equally gain and loss driven, whereas the N1 was particularly loss driven, whereas the N2 was distinct, not only it was lowest in HRD signatures, and it's also mutation driven. Now, the N2 stands out by having a distinguishable BRCA alteration patterns, and the N4 having distinguishable CCNE1 ATP gains. Whereas in the N3, furthermore stands out with low prevalence of TP3, 53 mutation, and late chromosome 17 loss. And then we see a PIK3CA and ARID1A mutations. So, and then in summary, timing of copy number and mutational events provides a deeper insight into how the tumor develops its genomic score, and this approach contributes to a refined subtyping of ovarian cancers. And such detailed analysis is only possible through the high-resolution data provided by whole genome sequencing. And these findings highlight the power of genomic event ordering and understanding ovarian cancer evolution and potential implications for personalized treatment strategies. With that, I want to thank you. First and foremost, a big thank you to Genomics England participants who have donated their samples and allowed their genomic and treatment data to be shared for scientists like myself to study. Their contribution is invaluable to our understanding for improving future treatments. And thank you everyone for listening.
Thank you very much, Aliah. Well, we'll just wait for some questions to come in, so please do use the chat box below. But I just want to say it was a really fascinating talk. It was really interesting to see the kind of distinctness between all the different groups. I was kind of wondering what your next steps were with the data, if you're planning to do anything extra to deep dive into anything more specifically or anything along those lines.
Yeah, this is just the start of some disciplinary analysis. There will be some deep dive, particularly in looking more into the clinical data available in Genomics England and see how everything matches or how everything links. I was hoping that we would have some response to chemotherapy data, but I don't think that's in gel, but it will be really interesting if we have that kind of data. Yeah.
Brilliant, thank you. And we've got a couple of questions by a question in the chat. So from Francis, thank you very much. Are you able to see any germline polymorphisms that predispose to any particular pathway, or would that not show up in your analysis?
I haven't looked at it yet. So by just doing the timing analysis, it hasn't showed up yet.
Okay, thank you. And then another one from Kitty. So thank you for the interesting talk. Are there any associations between cancer treatment and the 4 evotypes?
Yes, that's one of the clinical data that I will delve more deeply in later, because we want to see what the clinical data available in Genomics England. But yeah, interesting. Yeah, that's one of the next steps that I was planning on too.
Yeah, it sounds like you've got some more deep dive into the data that you can do, which is really exciting.
Yeah.
Brilliant. Can I ask as well, what do you think the impact this will be? Well, obviously at GEL, we're really interested in impacts for patients and things. What do you think? I mean, obviously there's more to do, but what kind of things hopefully will you get from the data in terms of impact for patients?
Well, I think it's about the treatment and how we can do some personalization and see why some treatments work on patients that we know maybe has HRD, but some doesn't work. So we could have, so the impact would be more personalized treatment, more effective treatment, more targeted treatments. That's what I hope to. But
yes, it would be
very
brilliant.
early.
Love for science to have a world-breaking impact, wouldn't we? And then another question in the chat box. So Danielle's asking if you've looked at the methylation.
No, I haven't looked at methylation, but interesting. Yeah, we'll have a look.
Brilliant, thank you. So if anyone else has got any more questions, please pop them in the chat. We do have a little bit of extra time here. So when in the presentation you were talking about DN2 and 4 were also slightly different. I know they had similar tumour types within them. Was there any differences in the tumour types between the two that kind of reason as to why they were so distinctive?
In terms of histological subtypes, no, there were not much difference of the distribution of histological subtypes. Both have really high grade series distribution between the two. So no difference in histological subtypes. It's just different within those cohort have different pattern of genomic events ordering.
Very cool. Thank you. Oh, we have one extra question. Can you elaborate on what version of cosmic signatures were extracted? Have you had a chance to compare your cosmic signatures with the recently published cosmic version 3.3 signatures profile in pan-cancer analysis? Sorry, it's a long question. I can repeat if you need me to.
Right. So I believe that is from 3.3. The latest version is 3.4. So, and those mutational signatures is actually the one that we've extracted for pan-cancer signatures, and we have preprints of that we've extracted mutations for a whole 10,000 or more samples from the Genomics England's cohort. So that's from the 3.3, and that would include the 3.4 version as well, because we've analyzed the signatures and introduced the new signatures that was included in 3.4 now.
Brilliant, thank you. And we've got another one. At what stage, with respect to early to late stage, is the mutational signature analysis performed? Did you expect any therapy-associated signatures popping up specifically across the DNs? In brackets, nice talk.
Thank you. Did you repeat the question? I didn't get the first part. Then.
At what stage, with respect to early to late stage, is the mutational signature analysis performed? Did you observe any therapy-associated signatures popping up?
All right. Okay. We didn't perform the extraction signature based on early, or so I didn't, or maybe I didn't look at it specifically whether those samples were at early stages or at late stages. So yeah, I haven't looked at it, but that's interesting perspective. I would try to look at it.
Brilliant. Cool. Thank you. Well, I think that's all the questions, and lovely job answering those. Thank you very much, Aliah. It was a really interesting talk. But I think we'll now pass to our next speaker, Claire, who's a PhD candidate in Dr. Bose's group in the Department of Pathology in Leiden University. Her focus is on copy number alterations in ovarian, endometrial and breast carcinoma. And Claire will be talking to us about reconstructing the continuum of copy number high endometrial carcinogenesis. The floor is yours, Claire.
I'm muted. Thank you for the kind introduction and, of course, the opportunity to share our work here. Aliah and I were actually colleagues because I did part of my PhD trajectory at the University of Manchester in the group of Professor David Wedge. So you may find some similarities in our talks. Before we delve into the results, I want to take a moment and reflect on the fact that it's September, so Gynecological Cancer Awareness Month. Also, with this study, we aim to contribute to the care of gynecological cancer patients, and specifically patients with uterine or endometrial cancer. Just to provide you with a bit of context, I looked up the UK statistics. So every year, over 9,800 women are diagnosed with endometrial cancer. So we're talking about 30 new diagnoses every day. And with that, I think the clinical need for this story is already quite clear. The way we look at endometrial cancer has fundamentally changed with this 2013 landmark paper. And after this TCGA publication, we moved away from seeing endometrial cancer as one disease, but started subdividing it into 4 distinct molecular subgroups based on the underlying genomic burden. On the left-hand side, we have the polymutants and microsatellite unstable endometrial cancers that are characterized by high levels of mutations. On the far other end of the spectrum, we have the copy number high subgroup that is chromosomally unstable and has high level of copy number events. This copy number high subgroup will be our subgroup of interest for today. And it comprises about 20% of all endometrial cancers, so a small subgroup. It is, however, a relevant subgroup for studying, because if we look at this Kaplan-Meier curve and the red line, this is the copy number high subgroup. So it is the endometrial cancer subgroup with by far the worst prognosis. And this prognosis remains to be poor, actually, despite the fact that we treat these patients quite extensively with the combination of chemo and radiotherapy in the adjuvant setting. Copy number high endometrial cancer is heterogeneous if we look at the clinical behavior, also at the histology, but they do have one defining hallmark, which is in the name, the fact that they're all copy number high. However, the fact that they're all copy number high does not mean that they're all the same on this copy number level. We can appreciate here 4 different copy number profiles. They're all copy number high, but they all look completely different. And this exact copy number heterogeneity has been the starting point of this study, in which we first aimed to identify distinct copy number genotypes in copy number high endometrial cancer, and with these genotypes to reconstruct the endometrial cancer carcinogenesis. Study design. I want to remind you of the fact that copy number high endometrial cancer is only 20% of all endometrial cancers. So we made some efforts and collaborated with different partners, both nationally and internationally, to find simply as many copy number high tumors as possible. With that, we had 2 different cohorts, a clinical cohort and a Genomics England cohort. And combined, we had over 3,700 endometrial cancers. The molecular classification has been performed for the majority of cases, and we selected from that all copy number high endometrial cancers. With that, we ended up with over 550 cases in the clinical cohort and over 140 in the Genomics England cohort. As a next step, we aim to capture the genome-wide spectrum of copy number events, and we therefore needed whole genome sequencing data. Since we only had formalin-fixed, paraffin-embedded material for the clinical cohort, we performed shallow whole genome sequencing with a horizontal coverage of below 1x. We were therefore very grateful for the opportunity to collaborate with partners in England, Manchester, Oxford, and got access to the Genomics England cohort that obviously includes deep whole genome sequencing data. So we have 2 cohorts, clinical Genomics England with shallow and deep whole genome sequencing data. And a remaining question now is, can we actually compare this in one study? To address this question, we downsampled the deep whole genome sequencing data of the Genomics England data set to obtain this shallow depth. We basically did a random sampling of 4 to 5 million reads. And with that, we disregarded over 99% of the remaining sequencing reads. With this approach, we were able to make a matched comparison of shallow and deep whole genome sequencing data. In terms of quantifying copy number events, we showed a strong positive correlation between deep and shallow whole genome sequencing data, with a Spearman R² of 0.89. And this actually gave us some confidence that we could compare deep and shallow whole genome sequencing data in one study, at least when focusing on copy number events. The starting point of the study was the clinical cohort, for which we performed shallow whole genome sequencing. We aimed to capture the spectrum of copy number events and therefore systematically deconstructed this genotype on 3 different levels. We first explored the segment size. We quantified the number of copy number segments, and we explored the fraction or percentage genome altered. And these 3 chromosomal instability related readouts were then used as an input to perform unsupervised clustering. With this clustering, we identified 2 distinct clusters that represent 2 distinct copy number genotypes. And these 2 genotypes have different patterns in terms of the chromosomal instability related readouts. And these patterns were not explained by technical features such as the predicted ploidy or tumor purity. 2 different genotypes. Genotype 1 in orange was characterized by limited numbers of long size segments. Copy number genotype 2 was characterized by extremely high levels of small size segments. And as a result, the fraction of genome altered on the right-hand side was actually quite comparable in the 2 genotypes. To refer to these 2 genotypes, we used the music terminology of tone lengths, long and short, longa and brevis. Here are 2 representative copy number profiles of longa and brevis copy number high endometrial cancer. On the left-hand side, the longa, we see indeed limited numbers of long size segments. And these segments are frequently confined to the entire chromosome or chromosome arm, as is the case for chromosome 7 and chromosome 10. In brevis copy number high endometrial cancer, we see a completely shattered genome with lots of small-sized events throughout the entire genome. So at this stage, we can already conclude that copy number high endometrial cancer comprises 2 distinct copy number genotypes. And for those 2 genotypes, we eventually aimed to reconstruct the endometrial cancer continuum, in which we focused on mechanistic, mutational, evolutionary, phenotypic, and finally clinical aspects. We have 2 different cohorts, the clinical and Genomics England cohort, and both cohorts have their own strengths, and we position the cohorts according to those strengths. So we started with mechanistic aspects using the Genomics England cohort. Longa copy number high endometrial cancer showed extremely large segment sizes. And these segment sizes, in terms of base pairs, actually approach the size of an entire chromosome or chromosomal arm. And for this reason, it was very likely that chromosomal misaggregation during mitosis has shaped this copy number genotype. This was a completely different story for brevis copy number high endometrial cancer. That seemed to be a composition of different events, including whole genome doubling, homologous recombination deficiency, chromoplexy, and actually the most striking difference between the 2 genotypes was the presence of chromotripsis. As a next step, we explored the mutational spectrum in the clinical cohort. We performed multivariate logistic regression, showing that individual mutations were strongly associated with one of the 2 copy number genotypes. Specifically, in the presence of a P10 mutation, a copy number high endometrial cancer was more likely to be longa. In the presence of a TP53 mutation, an endometrial cancer was at least 4 times more likely to be brevis copy number high. And this association is corrected for other individual mutations as well as the histology of the tumor. Following the strong enrichment for TP53 and PTEN, we further hypothesized whether different evolutionary trajectories underlie these two different copy number genotypes. So therefore, in the context of GEL, we performed evolutionary analysis using Placket-Luce, and you already had a fantastic introduction by Aliah. So what we did is we reconstructed the chronological ordering of different mutations in the two genotypes. On the x-axis, we see timing. So at the end of the plot, time of sampling, and towards the left, we go back in time. And the y-axis visualizes all of the different events. And I know that we cannot read all of the events from the y-axis as for now. So we're going to zoom into the earliest events of carcinogenesis. We identified remarkably similar trajectories in Lunga and brevis copy number high endometrial cancer, which was actually surprising to us. And the key differentiator, again, seemed to be the presence and frequency of PTEN mutations. So from this, we can conclude that it's likely that copy number high endometrial cancer starts on a certain trajectory, but upon requiring a PTEN mutation, it's more likely to become Lunga. Up until now, we only focused on genotypic aspects. And as a next step, we aim to explore where there's a genotype to phenotype relationship in the clinical cohort. So here's our plot again. So now corrected for different individual mutations, we see a strong association with histology. Specifically, the Lunga copy number high endometrial cancers were dominated by endometrioid histology, whereas the brevis were dominated by non-endometrioid, including serous histology. I hope I can help you with some visualization. So here are two Lunga copy number high endometrial cancers that look quite similar if we look at the genotype, but they look completely different under the microscope. The other way around, now we have two endometrioid copy number high endometrial cancers that look comparable under the microscope, both low grade, both endometrioid, but completely different on a genotypic level. So although we see a strong genotype to phenotype relationship, this is definitely not one-to-one. We know that histotyping by pathologists can sometimes be subjective, and we therefore aim to objectify this genotype to phenotype relationship by assessing nuclear atypia. Quantifying nuclear atypia by eye is not a good idea, so we leveraged a deep learning model that was able to automatically quantify nuclear atypia in a three-step approach. The input was a whole slide image, H&E, and all of the cells were segmented. And we're talking about hundreds and thousands of cells per slide, actually. After the segmentation, all of these nuclei were then classified as being tumor, immune cell, or normal nuclei. And in the final step, we gathered all tumor nuclei and looked at the nuclear size as a proxy of nuclear atypia. With that, we were able to show that brevis copy number high endometrial cancer showed a significantly larger nuclear area compared to the Lungas. In a similar way, we leveraged another deep learning model that has been developed by colleagues in Switzerland and is able to quantify mitotic counts. The input again is a whole slide image, and from that we selected the geographical area with the highest mitotic densities. And also in this context, brevis copy number high endometrial cancer showed significantly higher mitotic counts compared to Lunga copy number high endometrial cancer. So with that, we were actually able to show a genotype to phenotype relationship on three different levels: histology, nuclear atypia, as well as mitotic counts. The final step in the continuum is the clinical behavior of these tumors. And we did that in the context of the clinical cohort. Despite the fact that these Lunga and brevis copy number genotypes looks completely different on a genotypic and phenotypic level, the overall survival is actually quite similar. You could argue that all of these tumors were treated differently, which of course affects overall survival. And therefore, we reperformed the analysis in a population that is treatment naive and didn't receive any adjuvant treatment. And also in this context, we see a similar but poor overall survival. So they both have the same survival, but that does not necessarily mean that they have the same response to therapy. We had the opportunity to use the PROTECT-3 specimens in which endometrial cancer patients have been randomized into one arm, radiotherapy, or another arm, radiotherapy in combination with chemoradiation. And with that, we could really dissect the benefits from chemotherapy. Although the interaction p-value was not significant, we showed that Brevis copy number high endometrial cancer had a significant benefit of the addition of chemotherapy, which was absent in the Longas. And this is actually relevant information because currently both patient populations are treated with chemoradiation. In conclusion, we showed that copy number high endometrial cancer comprises of two distinct genotypes with two different copy number rhythms. The genotypes are shaped by different underlying mechanisms and mutations, showed profound histopathological correlates, and could potentially benefit from different treatment strategies. I want to thank everyone involved. It has really been a team effort. And I want to specifically thank the Transportec and Genomics England consortia for the uses of the data and material for this study. And happy to answer any questions.
Brilliant. Thank you so much, Claire. It was a really interesting talk and very, very well explained. We have one question already in the chat, so I'll just pop to that. Anyone else wants to pop their questions in, please do. So you have a question from Sophie. Was the extra chromosomal DNA looked at in these samples? Curious to know if the number of CN are related to the number of ecDNA in the cells.
Yeah, thank you for that question. And I do share this curiosity. I'm very happy to see what comes out of that. So I think that's for future work, definitely.
Brilliant. Do you have any kind of other plans for your future work as well? Just kind of what you think you want to do next with the data?
Yeah, I think we have a couple of avenues that we want to explore. I showed you that the Longa copy number high endometrial cancers do not seem to benefit from chemotherapy. So we would love to explore different therapeutic strategies for this subgroup. We already saw some hints towards different strategies, so that's something we're currently working on.
Yeah, it's really exciting that it might be able to shape more specific treatments for people. We have another question in the chat from Francis. So thank you very much for this talk too. As with the first talk, are you able to see any germline polymorphisms that predispose to longer versus brevis or to high copy number, or would that not show up in your analysis?
Yeah, thank you. I think that's a very relevant question. As for now, we haven't specifically looked at this. We do see that homologous recombination deficiency seems to be somewhat enriched in the context of brevis copy number high endometrial cancers. So we're very interesting to explore whether germline variants in BRCA1, BRCA2, or other homologous recombination related genes underlie that genotype, but that's something we're still working on.
Thank you. And just one more for me if we wait for some other questions. So I really liked how you looked into the shallow versus deep sequencing and just to make sure that they were actually comparable. I know you mentioned that you downsized the deep sequencing to get a random sample. I was just wondering if you did that multiple times just to make sure it wasn't just that one specific down sample that nicely correlated, or whether it was consistent, or whether there's anything else you did in your analysis to kind of check whether that variation you saw between the two distinct CN genotypes was accountable by the fact that it was either shallow or deep sequencing.
Yeah, that's a good question. So we downsampled all 142 copy number high endometrial cancers. We saw a very strong correlation. We didn't see any significant outliers in this context. But I want to stress that we didn't only look at copy number, the quantification of copy number events. We also looked at the other parameters, such as the fraction genome altered, as well as different copy number signatures. And for the majority of those, we see a strong correlation.
Yeah, lovely, nice. Thank you. Okay, we've got a couple more questions coming in. So one from Laura, great talk, thank you. 2 questions if possible. I'll do them one after the other so you don't get lost. First one, you showed different outcomes for patients treated with either chemo, radiation or brachy. Did you do any multivariate analysis adjusting for stage?
Yeah, that's a good question. So in the Protect 3, we don't have stage 4 patients, so it's limited to stage 1 to 3. But unfortunately, we don't have enough numbers to do multivariate analysis.
Brilliant, thank you. And the second question, knowing Brevis AUC patients had different outcomes, do you see this being used as a predictive marker? Is it a feasible test?
Yeah, we do think it's a feasible test. It's shallow whole genome sequencing. It can be performed on formalin-fixed paraffin-embedded material. There's no need for good quality DNA, and at least if the cellularity is okay, at least 30%, you can perform shallow whole genome sequencing. So in terms of feasibility, I think it's quite a feasible marker. So we'll see.
Brilliant, thank you. Okay, another one from Elizabeth. Thank you for the very interesting talk. Do you think the longer group is likely to be a subgroup of the no specific molecular profile group that we currently use in clinical reporting?
Yeah, so interesting question, a very valid point. We took a different angle. We started with the TCGA subgroups, so not a surrogate marker approach. So we started with all copy number high tumors, and you're correct, some of the longas, but also some of the brevis include NSMPs. We see in the brevis copy number high endometrial cancer a strong enrichment for TP53 mutations, but it's definitely not a one-to-one. And the final aspect I want to add, if we look at the Kaplan-Meier curve, they have the same overall survival, and that's not the overall survival curve we know from the NSMP subgroup.
Brilliant, thank you. Okay, another question from Anonymous. Thank you. Have you looked at mutational signatures and how these may differentiate longa and brevis?
Yeah, that's also a very relevant point. We're currently working on this, so to be continued. What we do see, I think in line with what we just showed, is that HRD is enriched in the context of brevis, whereas, for example, copy number signature, the Steele methodology, copy number 1, associated with the diploid genotype, is enriched in the longas. But we'll report on that in a later stage.
Brilliant, thank you very much. And thank you very much, everyone, for all those brilliant questions. That's really helped to stimulate some good answers from both the attendees. So I just want to say a big thank you to the brilliant speakers. I thought both the talks were really great and very informative. So thank you both very much for coming, and thank you everyone for getting involved with your questions. Please don't forget to sign up for the training, the RE training in November. We're really good to see as many people there as possible. And yeah, we really look forward to seeing you in the next seminar in October. Thank you very much for coming, and the speakers as well. And we'll see you next time. Thank you, everyone.